Proteins are the future of medicine, and they can be designed to treat infectious diseases, cancers, and diabetes. To create these protein-based therapies, we get our protein and create lots and lots of versions of it, and then we see which ones can bind or hug a target tightly. So why is hugging tightly the goal? Well, imagine someone getting tackled in football. Our proteins kind of do the same thing, where if they can bind or hug a troublesome target, it can be slowed or potentially stopped, and that can be therapeutic for the patient. So some of the targets that lie on the cell surface can't be targeted, and they're termed undruggable. Why? Well, there's a lot of structures on the cell surface, and to design a drug that's just specific for just that target, we must isolate that target from the cell itself. And methods to do this are working on a case-by-case basis while being time-consuming, complex, and/or expensive. So this is problematic, right? As the cell surface is a hot target when dealing with multi-drug resistant bacteria, nasty viruses like the one that caused COVID-19, and even us. So, what if I told y'all that I'm working on a technology to bypass the need to isolate this target? I plan on using the cell itself to help me engineer medicine. To create these protein-based therapies, I'll start by making, of course, lots of protein. I will attach DNA tags to each protein. The DNA tags serve as a label to reveal the protein's identity. I'll also attach DNA tags to the cell surface protein of interest. The key is that I'll modify both the DNA attached to the cell surface target and the proteins in a way that makes them perfect for each other once they meet. Romantic, right? Once there's an interaction, a unique structure forms, and a signal is produced telling us that this is a protein that hugs our target tightly, and we want to keep it, while the other proteins that hug the cell but not that target won't produce a signal. So far, I have the unique structure, I have the DNA tags, and the signal all figured out, but these are proof of concept experiments done without the proteins or the cells. But I plan on getting much closer, however, by attaching DNA tags to many proteins, and then looking for which ones can make this signal by binding to a target tightly, while facing competition from a lot of others that don't. In the end, my technology will allow for proteins to be the go-to when dealing with undruggable targets on the cell surface. And as more drugs are made and tested, the chances of one being approved decreases. And these very special proteins will help treat human diseases relating to undruggable targets. Talk about a game changer. Thank you.